Hi, everyone. So I decided to go ahead and do a little like mini review video for the final. I will be posting all of these slides onto Canvas for all sections. And then I'll also be posting this like mini review where I'm going to touch on some like the major concepts just to give you guys something to kind of listen to and follow through as far as, reminding yourself about some of the stuff we talked about in past chapters that we haven't seen for a while. And the other part is we're going to, for those of you who have in-person class and not the online group, my traditional groups, you guys, we're going to be having a Kahoot in person, like a live Kahoot in person to review some of the stuff for the final. I'm going to put like a separate Kahoot quiz together for that live. So if you guys are my online students, you are welcome to join in with that. I have in-person reviews that will be going on that'll be this Wednesday or Thursday, and that'll be Wednesday at 10 a.m. or it will be Thursday at 10 a.m. or Thursday at 5:45 p.m. You're welcome to join in on Zoom, or you are welcome to, I guess, join in person, of course, if you are available at those times. Anybody in any of those classes can come to any of those times actually to participate if you want to. And then I will probably record one section, not all of the sections, but one section maybe all of them. I don't know. I haven't decided, but at least one, and then posting that like Kahoot session on the Canvas sites. So maybe all of them. We'll see. So right, so let's just do the big like basic rundown of like the major topics of the stuff that we talked about all throughout the entire semester. It's a lot, so I'm not gonna be able to cover everything, and we're gonna be skipping some of the stuff on the slides. I just want to be sure that we get like a brief video, about 30 to 45-minute video in, that you guys have something to refer to. So let's do that. Okay, so starting with unit 1, we had chapter 1 was about the basics, right? We talked about how microorganisms are just anything that is an organism small enough that you need a microscope to see it, and that pathogens are disease-causing microorganisms. If you don't know about the steps involved in gram stains, if you don't know what an acid-fast stain is by now, I'm a little concerned probably. So remember, mycobacterium will be the ones that probably will be the most concerned about as far as acid fast is going to go. And endospore is also a very important concept. And we'll be talking about those a little bit when we get into the prokaryotes. So don't want to get into too much of an issue here. But remember, the endospores are not metabolically active. They are metabolically inactive. So they can survive very harsh conditions. Apparently we're going to talk about chapter 3 before we get to chapter 2. That's fine, right? So chapter 3 was talking about the tools that were involved in microbiology. That includes our 5 I's. Those are basically just the steps that are involved in trying to investigate what an organism is when you're trying to figure out, what's the causative agent of, your patient's illness or whatever it is. So inoculation and incubation. So it's like literally just like taking a swab off of the bottom of your shoe and spreading it onto just some random agar, incubating it. And then you can take samples from that to start with whatever isolation methods you want to use, whether it's dilution or streak plates or something like that, right? Inspection means running tests like all of our physiological tests, like the citrate test and the malonate test and all of those kinds of things. And then identification is using the information that we got from all of this to identify whatever the organism is. So those are the 5 I's. Don't forget what that is. Selective media, certain things grow on it, certain things don't. And then differential media in general, it's not going to be necessarily selective. It can be both, but then it is selective and differential, right? So differential is just allowing you to tell the difference between maybe different metabolic or physiological aspects of the bacteria that are growing on there, usually based on a color change or some sort of change, visible change in the media, right? Fastidious is a term referring to organisms that have to require growth factors. So they are kind of picky eaters, and growth factor is something that you would add into the media that the organism can't produce for itself. It has to have it in the food, like there already. we talk about pure cultures. That's just a culture that represents like one bacterium that's been isolated and grown out. Yeah, not from one source, because you can get a lot of different bacteria from one place, right? Like the bottom of your shoe. So just culturing that up wouldn't be a pure culture. It has to be one species. We talked about this. You guys know, man, the freaking microscope. You should know that by now too. So chemistry. Sorry, I keep clearing my throat. I had to decide right before I filmed this to drink Soylent. And Soylent, I mean, it's basically plant milk. So it's like sticky in my throat. It was a bad idea, but I do want to get this filmed. So chemistry, we talked about the different kinds of bonds. Covalent bonds share electrons. Ionic bonds, you have a transfer of electrons. Hydrogen bonds have to deal with one atom hogging the electrons from the hydrogen. Typically, hydrogen gets a positive aspect to it. Other molecules that have a negative aspect to them will, because they hogged electrons. So the negatives and the positives will be drawn to one another, and that's a hydrogen bond. There's no exchange of electrons necessarily going on there in that bond, okay? Organic molecules are carbon and hydrogen based, okay? pH, 7 is neutral. Anything lower than 7 is acid. Anything higher than 7 is a base or alkaline, okay? We talk about the protein structures. The amino acid sequence, just the straight up amino acids that are in a protein, because that's what proteins are made of, by the way. That sequence represents the primary. The secondary is going to be made up of the structures that are going to be involved in the associations of those amino acids with one another, the nearby ones. So that'll form things like beta sheets and alpha helices. Tertiary is going to be, things like the disulfide bonds and the overall folding of that peptide. And then quaternary would be like polypeptides coming together and associating to create a protein that works. All right, prokaryotes, these are like bacteria and archaea. They don't have any internal membrane bound structures, okay? They have a membrane that encloses their inside. Then they have a cell wall. Some of them have another membrane outside of that, right? So that's that whole structure, membrane, wall, and sometimes another membrane. That is their cell envelope. Peptidoglycan makes up the cell wall of bacteria. It's made up of NAM and NAG, N-acetylglucosamine and N-acetylmuramic acid. I don't need you to know what that means, but that is what that is, okay? And then they are cross-linked across one another with peptides, okay? And gram-positives, lipoteichoic and teichoic acid create some support and structure and help during, a binary fission whenever the cells are dividing. Gram-positives have a thick peptidoglycan wall. That's where the gram stain comes into play, right? So the crystal violet gets stuck inside of that thick peptidoglycan wall. And when we force that to stay in there using iodine, that sort of cross-links across everything, and acts as a mordant to keep it in there. Gram-negatives has such a thin peptidoglycan wall that the crystal violet's not going to stay in there. So it just rinses out with the alcohol step, okay? So that is what's going on with the gram-negatives. So that is why you will counterstain with safranin, and the gram-negatives are red and the gram-positives are purple, right? Acid-fast, that's for bacteria that have mycolic acid in their outside. That was the mycobacterium. And mycoplasma, they don't have a cell wall at all, okay? What sorts of cell shapes? Cocci are round, bacilli are rods, and the arrangement, staphylo, the grapes, strepto, the chains, and diplo is pairs. We move around as bacteria. We move around with these things called flagella, okay? You can have polar flagella, amphitrichous, so at both ends. You could have like tufts at both ends. Monotrichous, so you have just one flagellum, and then peritrichous, where it's all over them, like all over the outside. Capsules kind of allow for sticking as well as evasion of phagocytosis. Slime layers, which we didn't talk much about, but that also has a similar effect. And then the development of biofilms, where the bacteria will kind of grow in mass and signal with one another to tell each other about, the environment around them and how everybody's doing in that whole mass, essentially. And that's called quorum sensing. Endotoxin is part of gram-negative outer membrane. So the gram-negatives, because they have that thin peptidoglycan wall, they also have that outer membrane. There's something in their outer membrane called lipopolysaccharide. Part of that is endotoxin, okay? That's where that comes from. It's only gram-negatives. Plasmids are little small circular bits of DNA. They are not essential for survival. They can code for virulence factors. Remember, prokaryotes too, they have a single circular chromosome, much larger than plasmids, but it is a single circular chromosome typically. And then we have endospore formation. Vegetative cells are the normal free-living metabolically active cells. The endospore itself is the metabolically inactive, protective version. The process from going from a vegetative into an endospore, let's say you're running out of some sort of nutrient, like a carbon source, you will go through something called sporulation. That's forming the spores, okay? And then sporangia is the cell that's surrounding, like basically, let me draw it out. If you're a cell, oh, okay. Sorry, this was not what I was expecting to have happen. Let me pull this up. Go back. Okay. If you are a cell and you're happy and you're vegetative and you're free living and you're metabolically active and you run out of a carbon source, and so you want to make an endospore, right? So you start forming an endospore, I don't know, at one end, whatever. Here's your endospore forming right here. And it would stain green, right? With what stain? Malachite green. That's right, with the steam forcing it in there. So, and then we counterstain. And what cells or what parts of the cells would turn red? It would be the vegetative cells. In this case, this portion out here that's surrounding the endospore, which is the dark part that I filled in, that's called the sporangium. Okay. The sporangium is that part that is like left of the original vegetative cell while it's trying to make an endospore. So that's the sporangium. Sporangia is plural. And then germination, that's whenever we have that endospore and now it's found its carbon source later on, and now it's going to start turning into a vegetative cell again. So it'll break out of its little, covering, its cortex, its shell, and turn back into a vegetative cell. That's germination. Okay, eukaryotes. This is like you and I, we are eukaryotes. They have membrane-bound organelles. This will include, animalia, which will be like helminths and the flukes and all that sort of stuff. I did want to say that the Protista kingdom, that has to deal with the algae and the protozoa. So don't forget about algae, okay? And remember, algae are photosynthetic. And that most of the photosynthesis that happens on Earth to create the oxygen that we breathe, that happens thanks to microbes, not plants. Okay. Okay, right. So organelles, they are membrane-bound things inside the cell that have like some sort of function, right? Prokaryotes, they don't have that. They have ribosomes, but that's not an organelle technically, right? That's just RNA pieces that are folded up that do some work. So what are some of the organelles that you should probably be aware of? The mitochondria, that's where aerobic respiration happens, where the electron transport chain is. That's where energy is made. It's the powerhouse. The endoplasmic reticulum, where we're going to be doing translation. The ribosomes kind of stud the outside of this, and we make proteins, and they'll get processed in the endoplasmic reticulum. Then we have the chloroplasts. The chloroplasts are for photosynthesis. The nucleus, that's where we're going to store our genetic information. And we have the Golgi apparatus, which does some more modification and packaging. So that's not on there, but that's a thing. So trophozoites are protozoa that are free living. And then they have their own form that is not the free living form, called cysts. And these are the protected forms. They're very similar to endospores for bacteria. Viruses, these are obligate intracellular parasites, meaning they have to live inside of cells. Their genome can be single or double-stranded, RNA or DNA for both of those. It can be any of that. That genome is then coded in a capsid. Capsids are made of something called capsomeres. Those are just units that come together to form the capsid. The capsid is the protein coating that surrounds the genome. That's really all it is. They can be icosahedral, made up of triangle shape, or it can be helical, which is exactly what it sounds like, right? So wrapping around it in a helix shape. They have spikes on their outside. Those spikes are glycoproteins that interact with host glycoproteins, host surface markers, okay? And that interaction is what allows the virus to get into the host cell. Some viruses have envelopes. That just means they stole a membrane from their host whenever they were, budding out of the host cell. So, yeah. Right. Bacteriophages are viruses that infect bacteria, right? They always lyse their cells. They don't bud out. They always just break out. Latency and lysogeny. Lysogeny is basically the bacterial version of latency, but lysogeny refers to the bacteriophage genome being incorporated into the host DNA. And then the lytic cycle is whenever the normal viral cycle is going on. We're replicating out in like the cytoplasm, and the genome isn't hiding out anymore, okay? Oncoviruses cause cancer. They're cancer-causing viruses, right? And viruses are very simple. They are not cells. They are not alive. They lack organelles. They lack ribosomes. Getting into unit 2. All right. Growth preferences. We had the saprobes that break the dead things down, halophiles that grow in salt, thermophiles that do well in high temperatures. Symbiosis, that means whenever organisms live in a close association with one another, and their lives can often depend one way or the other on another organism that way. Commensalism is when one organism benefits and the other is whatever, it doesn't get affected. Mutualism, both organisms will benefit. And then parasitism, one organism benefits at the expense of the other, which is the host. The host is getting damaged by the parasite. All right, making energy. This is where we were talking about metabolism. This is where we get into aerobic respiration. This was a very, very long chapter. It's too much information, I think, for one chapter, but it is what it is. So catabolism, cats tear things apart. Catabolism, breaking molecules apart to store energy, right? You break things down into small parts, you are releasing the energy that was stored in their bonds. So now you can, basically store that energy or use that energy or pair it to other reactions. So catabolism is breaking things down in order to get the energy. Anabolism is making larger molecules. You need energy in order to do that. I dare you to go build a whole building without energy at all. So you need energy to do those things. Enzymes, these are catalysts. Those are things that help lower the activation energy, so the amount of energy required for any sort of chemical reaction to happen. Enzymes will help lower that, so they can happen more easily and with less energy going into them. But the enzyme themselves won't be permanently changed or used up. So the thing that the enzyme acts on that is causing it to change, from A to B or something, or like breaking something apart, whatever it's doing, that is called the substrate. That's the thing that's being acted upon. Some enzymes require the presence of like helping factors in order to help them do their job. Coenzymes are basically organic molecules that would help out an enzyme or bind in another area of the enzyme to allow it to do its job better. Competitive inhibition, that is anytime something that inhibits that process from happening, whenever that inhibitor that's doing the inhibiting it interacts with the actual active site where the substrate would bind, that's competitive. It's competing with the substrate. Non-competitive inhibitors, the way that they work is they bind on another area of the enzyme that can cause often a change in the shape of the enzyme. And then whenever that change in shape happens, it can no longer interact properly with the substrate at the active site where it is not interacting. But you get the idea. Okay. Aerobic respiration. This is how we're going to make energy whenever you can have oxygen available. Because why do you need oxygen? Because it is your final electron acceptor in the electron transport chain. Okay. All right, glycolysis, the Krebs cycle, and the electron transport chain. Glycolysis, we're going to break down glucose into pyruvate, essentially 2 molecules of pyruvate. Pyruvate is a 3-carbon molecule. Glucose is a 6-carbon molecule, okay? So breaking down that 6-carbon glucose into 2 pyruvates. Now, those pyruvates are going to be converted into acetyl-CoA, and then that's what's going to enter into the Krebs cycle. And essentially the Krebs cycle, which is also known as the citric acid cycle. essentially what's going to be going on there is we're going to be breaking some bonds and moving around some electrons, or we're going to have carbon dioxide made as a waste product through this. And during all this switching around, we can take some electrons and put them onto electron carriers. So that would be NAD and FAD. So those guys become NADH and FADH2, and they're carrying electrons along with those hydrogens. Carbon dioxide is given off as waste during this point, and this is why you exhale carbon dioxide. Okay, so now we've got NAD and FADH, and I just told you those are electron carriers, and the electrons are associated with the hydrogens, right? So we're going to shuttle those through the electron transport chain. And what's happening there is we're taking the electron and pushing it through these other molecules called cytochromes. And as we're doing that, it causes protons. So, I mean, where did that come from? Well, when you had NADH, we made this in the Krebs cycle. Basically, what happens is we have NAD, we have H, and we have electrons. So it's still the same thing. It's just broken into its little pieces. So this guy is going to go re-enter the Krebs cycle and go pick up more electrons, right? Well, our protons, which are hydrogen atoms without electrons, and then our electrons, these guys go through the cytochromes, and these guys form the proton motive force. And basically, they go outside of the membrane, they get pumped out as the electrons move by. Electrons are shuttling through, and then that forces the protons out, 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 out. So we build up this gradient of protons outside of the membrane. All they want to do is come back inside and equal out that, like what we would remember about diffusion, right? And osmosis and all of that. So, which I didn't mention in chemistry, but you might want to, review that a little bit if you aren't familiar with it. So, right. So those, we've created a gradient, and now those hydrogen ions, those protons, want to move back into the cell. The only way back into the cell is through this thing called ATP synthase, which is an enzyme. And when the protons move through back into the cell, it powers the phosphorylation of the ADP to turn it into ATP. And ATP is what the cell will use for energy. It basically clips off that third phosphate of adenosine triphosphate. And then that releases a ton of energy that the cell can use for whatever thing it wants to use. And then we have our leftover with ADP, and we can go back and send it back over here to get another phosphate added on over and over and over again. And that's how all of that will work. So that's the idea of what aerobic respiration is all about. There's a lot more information about it that I could go into, but that's the gist of it. All right. This is all happening in the mitochondria for eukaryotes, okay, on the inner membranes of the folds of the matrix in the mitochondria. In prokaryotes, like bacteria, this is happening on the inner membrane, like of the cell, just on the inside of that, okay. All right, fermentation, when oxygen isn't available, facultative anaerobes can switch between aerobic respiration and fermentation. So we'll just use glycolysis in this case to make certain byproducts like alcohol and acids, and then in that meantime make ATP very slowly through that. Strict aerobes can only do aerobic respiration. That's it. They have no other alternative. That's like us, right? So if we don't have oxygen, it doesn't work. So facultative anaerobes, they can ferment. Obligate anaerobes. So these are these are organisms that they cannot be in the presence of oxygen because oxygen is associated with toxic byproducts. So they don't have the means to deal with that. So they can actually die from oxygen exposure, but they have other molecules they'll use as their final electron acceptor. Photosynthesis, here we have the light cycles that will use energy from sunlight. We'll capture that sunlight energy to make ATP, pretty similar to the electron transport chain. Instead of using electron transport, now we're using photons. And they will make some ATP, just a little bit, And that's enough to power the chloroplast so that it can fix that carbon from carbon dioxide from the atmosphere and then put it into glucose. And then we make oxygen as a byproduct of that. And then they can use glucose for aerobic respiration. All right, genetics. DNA is deoxyribonucleic acid. That's basically our chromosomes and our genetic makeup. This is going to be referring to any living thing. So not viruses necessarily, they can have DNA. They can also have RNA. It can be single-stranded or double-stranded for either of those. But if it's alive, if it's a living cell, then it has to have DNA and it has to be double-stranded. All right. So it's our chromosomes and our genes. The basic building block for DNA is the nucleotide, which consists of a nitrogenous base, a 5-carbon sugar. In this case for DNA, it's deoxyribose. For RNA, it's ribose, and then a phosphate group. RNA is ribonucleic acid because ribose is the sugar. It does a lot of stuff for the cell, including being like that messenger copy of a working copy of the gene of whatever you're trying to make, a protein of. For transfer RNA, for ribosomal RNA, and then there are some other functions as well, like microRNA and silencing RNAs. RNA does not use thymine; it uses uracil. So instead of T, it has a U. When you look at the DNA nucleotides, every 3 nucleotides, which we call a triplet in DNA, in the RNA messenger RNA version, we call that a codon, which is a CUG. Those will code for 1 amino acid. So the triplet of the DNA or the codon in the mRNA, that codes for 1 single amino acid whenever we go into translation by the ribosomes. Mutations can be missense. So if you went from like, I don't know, AAG and it became AAA and it did code for phenylalanine and now it codes for leucine. I mean, I'm just making this up as I go, but that would be missense. So you would change the amino acid from what it should have been. All right. Silent mutations is if you went from AAA to AAC and they both coded for leucine. In the end, the amino acid is the same. That's a silent mutation. Okay. The amino acid didn't change. If it changed, it's missense. If the amino acid changed from an amino acid to a stop codon and totally stopped everything, that's called a nonsense mutation. All right. We also have frame shift mutations, which you have a deletion or an addition of nucleotides that shift the entire reading frame and therefore cause huge, huge changes in the entire amino acid sequence of a protein. Restriction endonucleases are enzymes from bacteria, essentially cut DNA at specific sequences that tend to be palindromic, so same forward and backward, and create sticky ends that we can use for genetic engineering to stick things in, like specific genes into plasmids. We can insert those into bacteria and cause those bacteria to make whatever proteins from whatever genes we want. PCR is polymerase chain reaction. It's very, very, very small amounts of DNA and can amplify it into massive amounts in very short time, just a couple of hours. Steps will include heating. High heat denatures or separates the DNA strands from one another. Primers then attach on. So basically we have our normal DNA, like it's like a ladder, right? It'd be a helix, but you get it. And then we cause heat causes that to separate. And then we're going to add our primers with slight cooling here so that they'll stick a little bit, right? And then we will add Taq polymerase, which is a DNA polymerase from Thermus aquaticus bacteria. And then that will create essentially another DNA copy so that now that will become like this. And just like any other version of DNA replication, this is semi-conservative. You are separating 2 old strands apart, adding a new strand to each of those old strands. So you now have 2 strands. One of them is old plus new. The other one is old plus new. That's called semi-conservative. All right. During cloning, DNA can be introduced to target organisms via plasmids and whatever else. The other thing I want to mention a little bit about this is that whenever you are copying DNA and you will like open something up. So it's like, okay, here's your DNA, and then you go from, you're going to open it up because you're going to make copies of it. Now, imagine, I mean, this is a long, this is like our DNA, okay? Super, super long. So you're going to open this guy up. And now you've got one side over here and one side over there. The DNA polymerase only moves in the 5' to 3' direction. So if we start over here and we go 5', this is 5', let's say this just talks about the chemical, what's makeup of that end of it. That's the 5', and this is the 3' of that strand. Well, they're anti-parallel. They don't go in the same direction. So it's not like this is the 3, right? No, on that other strand, the anti-parallel strand, this is the 5 and this is the 3. So if you can only move in 5' to 3', you can make a little bit at a time with your polymerase, and this will open up more, and then you'll make more. But you see how now you've got little pieces? These little pieces here, we can join them, join the pieces by DNA ligase that seals the DNA together, okay? But the reason that you need to do that is because these guys don't just automatically do that themselves. They're little pieces, and those pieces that we've created here, those are called Okazaki fragments. All right, that's what I wanted to say about that. What did we talk about bacteriophage? Okay, well, yes, whatever. We're gonna talk about that more later. Okay, there are different climatic regions, so literally different climates, different terrestrial areas where we have different things that are growing in different areas of the world and all of this stuff. Those are called biomes. Okay, that's the term for that. In the food chain, in the ecosystem, the primary producers, they basically make their own carbon source and they make their own energy. So now we're talking about plants, right? They fix carbon dioxide, which is not an organic molecule. They fix it using sunlight energy to create glucose, so they can use that as a carbon source. They also utilize sunlight for energy to create more ATP later on, right? So that's how they do everything. They do it all from scratch. And then therefore they are producing all of that. So they are producers. So they are primary producers. Everything above them in the food chain relies on them for carbon source and energy. All right. Potable water is any water that's free of pathogens. This is consumable water. Food safety. Cold and freezing won't kill anything. You have to heat it up above a certain temperature in order to kill stuff. Cold will only suspend growth. All right, microbial control. Asepsis is a term for removing vegetative microbes from living surfaces. Decontamination is basically the same thing for non-living surfaces. Sterilization is complete and total elimination of all microbes, including endospores. Nothing survives. Microbistatic, we're suspending growth, but not killing them. Microbicidal, just like homicidal, this kills microbes, okay? We have physical, chemical, and mechanical means of removing microbes. The physical mechanical versions we talked about, radiation, we talked about ionizing versus non-ionizing. UV radiation, for example, that is a non-ionizing radiation. It's relatively tame and is not going to sterilize things, not completely. In fact, a lot of things have the ability to repair damage done by UV radiation. Ionizing radiation, on the other hand, can sterilize things. Ultrasonic waves can be used. Heat, moist is more effective. Moist heat is more effective than dry heat, but you can use either one. Incineration is common. We use that for our loops, right? Tyndallization and pasteurization, so cycling between heat and cool, and then to avoid causing damage to liquids. And then boiling, that is not going to sterilize anything, but it could potentially make something potable, right? And cold, just preservation, desiccation, osmotic pressure. None of this stuff is going to be sterilizing, but it is effective at slowing microbial growth. Chemical means of microbial control. Halogens, some of these can be used to sterilize. This is like the chlorines and the iodines. Phenols, alcohols, hydrogen peroxide, which can be used to sterilize. Surfactants and detergents will not sterilize, but they're usually used in conjunction with other things to help them become more effective. And remember that surfactants, they're going to disrupt the cell membrane because they look a lot like phospholipids. And so they can kind of squeeze in between the phospholipids, and then that causes weakening of the cell membrane and then leakage of the cell. And that can allow other chemicals to get in. So that's quite effective. Aldehydes, they can be sterilizing. We talked about a lot of these. Most commonly, when we talk about sterilization of like medical instruments, we're talking about autoclaves. They use heat, steam, and pressure to sterilize things. Incineration and filtration can also sterilize when used properly. Ionizing radiation can sterilize, but non-ionizing like UV will not sterilize. Right. I think that's pretty much all I want to say about that. Remember that tyndallization and pasteurization, like what we do with milk. Right. Milk, it ain't sterile. So pasteurization does not sterilize. All right. Drugs to treat infections. Selective toxicity refers to when you want maximal toxicity against the bad guy, the microbe, the pathogen, with very minimal damage to the host. So preferably no damage to the host. That's selective toxicity. Toxic to the bad guy, not toxic to the good guy, right? Antibiotics treat bacterial infections. Antibiotics are not used to treat viral infections. Antivirals are. So common drugs that you might want to know, here's the big ones. Cell wall antibiotics are going to be the penicillins and the other beta-lactams. Ribosomes, the ones that infect the ribosomes are going to be aminoglycosides, tetracycline, and some of the mycins. Folic acid are going to be sulfa drugs that includes trimethoprim. Nucleic acid blocking, or basically the synthesis blocking medications, we have fluoroquinolones. They usually mimic the nucleotides, and then that's how the whole process falls apart. Antivirals can include Tamiflu, which we talked about Tamiflu and how it blocks the function of neuraminidase. And then acyclovir, which blocks nucleic acid synthesis in herpes viruses, but can be used for other viruses, very widely used. Antifungals will include anything in azole, so ketoconazole and stuff like that. Antiprotozoals, we have the quinine-based drugs for malaria and metronidazole. You can also remember that we can use the Kirby-Bauer assay to find an antibiotic that would be useful against a specific bacterium. If you are unsure if your patient can be treated with a certain antibiotic, you can do that and determine the sensitivity of that antibiotic against that bacterium that you're looking at. All right, they're talking about infection and disease. Right. Your normal biota actually plays a pretty big part as far as protecting you via microbial antagonism. So again, taking up the stadium seats and producing any toxic substances that could prevent other things from coming in and growing that could be dangerous for you. As far as infection goes, we have whatever the portal of entry is for the organism. Organisms can be, pathogens can become coming from areas that are exogenous or endogenous, so from inside of your body or outside. Stuff that's already growing in your body can become a pathogen to you, can cause infection, so beware of that. Attaching an adhesion to the host, surviving host defenses, such as antiphagocytic factors like capsules, causing disease, that includes virulence factors like toxins, that also includes endotoxin from the gram negatives. Signs and symptoms, once we start causing disease, signs are going to be anything measurable, like a fever, leukocytosis. If we're having a lot of white blood cell production, you can measure stuff like that. Symptoms, these are going to be reported. So this can be like having a lot of pain or having nausea. We can't record that. We can't see that for ourselves. So that's just reported based on, what the patient is telling you. And then syndromes are diseases that we describe based on their signs and symptoms and other data, like meningitis or pneumonia that can be caused by a variety of things. Then we have the exit from the host and the phases of the disease process, including incubation, which is we're not going to have an increase in like the viral load or something like that until we start having the virus or whatever taking hold inside the body, right? Prodromal phase, that's when we start having the initial presentation of the symptoms. Invasion, where we have, exponential growth and increase of those symptoms and signs. And then convalescence, where we start seeing a decrease in the symptoms and the signs as the body recovers. We talked about reservoirs versus sources. So reservoirs where something is maintained in the wild, sources where it may have come from, versus carriers, which would be somebody that would just necessarily be carrying that thing. It wouldn't necessarily be a reservoir, but it's just carrying it in that circumstance. Zoonoses come from animals. As far as transmission, I do want you guys to be aware of the vehicle type of transmissions. So like fomites, where somebody touches a door handle or something, and then somebody else comes and touches it, and they got the virus from that previous person onto them. So that is when something is acting as a vehicle. That's a fomite. Nosocomial infections are infections you get from healthcare settings. I know a little bit about the difference between the terms involved in epidemiology. Prevalence and incidence, prevalence being like over a long period of time since the beginning up until now. Incidence is referring to maybe like a specific outbreak or for this month, and that sort of thing. Morbidity is how the sickness. Mortality is the death rate. And then there's the other terms. I'm not going to go through all of them, but. All right, non-specific immune response. This is our first line of defense and our second, okay? So the first line, things like the skin, lysozyme, and everything like that. Lysozyme breaks down peptidoglycan. It's present in tears and saliva and everything. All right, then the actual immune system itself, the non-specific immunity. This is the innate immune response. So this is the general immune response. Has to be able to, tell the difference between self and non-self, which it does via recognizing certain surface markers on cells. The lymphatic system includes the thymus, the lymph nodes, the spleen, and the lymphatic fluid. The white blood cells are going to include all of the white blood cells, not just the lymphocytes, which are the T cells and B cells. White blood cells are the leukocytes. We have the granulocytes, the most numerous of which are the neutrophils. The phagocytes, which are things that eat up the bad guys. So that is phagocytosis is any time that a cell comes around and takes up something from its environment and brings it into the cell and breaks it down. That is phagocytosis. So phagocytes are going to be neutrophils, monocytes, macrophages, mast cells, dendritic cells. We already said about phagocytosis. There's the steps of phagocytosis on here. This is going to be, I'm not going to read through all these, but I would be aware of the steps that are involved in it. PAMPs are pathogen-associated molecular patterns. This is how the innate immune system responds to a bad guy. These are things that it sees as automatic bad guy. Peptidoglycan shouldn't be hanging around in the bloodstream. Neither should endotoxin. Stuff like that, the body will automatically see that as bad guy associated. So those are PAMPs. The agranulocytes, the ones that don't have granules automatically in them, are going to be the monocytes, macrophages, and the dendritic cells. That also includes the lymphocytes. All right, as far as the inflammatory response, which is the number one response you're going to get from the innate immune system, rubor, redness, calor, warmth, tumor, swelling, dolor, pain. And then fever is really important in all of this. Fever is induced by pyrogens. Pyrogens don't just always come from bacteria or pathogens. They can come from signals from within our body as well. Sometimes those are, transmitted due to a pathogen being there. But yeah, it doesn't always come directly from pathogens. Endotoxin is a pyrogen that can lead to a fever. Remember that fever increases cell metabolism and activity, decreases microbial growth, releases transferrin, which keeps iron away from the microbes. And, it can be caused by exogenous or endogenous signaling. It's not always just bacteria or viruses. All right, interferon is something that we use to signal our buddy that we are getting infected by a virus. So that is a cytokine that does that. And then complement cascade, basically that is going to, we have classical, alternative, and the lectin versions. In the classical version, we have antibody that binds onto something, and then complement protein recognizes it. We have a cascade, which just means that, one protein signals the next to be cleaved or come together or whatever. That can lead to opsonization, cytolysis, or inflammation. Cytolysis is poking holes in things so that it leaks out. Inflammation, we just talked about what that is. And then opsonization, which is going to be helping with phagocytosis. With the specific immune response, here we're talking about our lymphocytes and their function. They will, at first, when they're activated, proliferate first, and then they will differentiate into memory, effector, or regulatory cells for each kind. Clonal selection early on in the process of the maturation of these cells, that should be getting rid of anti-self ones, so the ones that would react against yourself. So in order to get an immune response, you have to be a foreign molecule. You have to be seen as foreign. In order to be reacted with appropriately, you have to be big enough. So things that are too small are haptens. So they have to be linked onto something else for us to react to them. Super antigens induce overreaction by the immune response. This is where we get toxic shock syndrome. And then antigen presenting cells like macrophages, monocytes, dendritic cells, and even B cells themselves, they will have interaction with whatever the pathogens are and go and present them to like T helper cells to have them be activated, and that will, create the trigger of all of this whole response. So an APC comes along, interacts with a T helper cell. The T helper cell says, yes, this is the bad guy. I agree with you. And then activates the B cell or activates the APC that it's even interacting with to go fight more, or goes to interact a cytotoxic T cell, whatever is going on. So that's how that's all triggered down the whole line. MHC1 is associated with all nucleated cells. All nucleated cells, if it has a nucleus, it's got MHC1 on the outside of it. This is how they're going to be presenting endogenous antigens. So if you're infected with a virus, for example, whereas MHC2, that's going to be only on our antigen presenting cells. They're picking up exogenous, so stuff from outside of the cells, like from the bloodstream or whatever. Okay, T cells. Whenever our T helper cells are activated by MHC1, the endogenous, like internal viral, these are our CD4 T helper 1 cells that will eventually activate the CD8 type cytotoxic T cell response. They're going to kill bad guy cells, infected cells that have the virus or whatever in them, with perforins that poke holes in them and granzymes to help dissolve the things inside of the cell, and that will lead to apoptosis of the cell or the cell killing itself. With the MHC2 complexes, CD4, which is helper cell marker, CD4 T helper cell 2s will activate the B cells. When B cells are activated, they will become plasma cells that produce antibody. Right. So FAB is the variable region of the antibody. That's the part that actually interacts with the antigen. What does antibody do for us? Well, neutralization. So basically covering something so it can't be doing its job anymore. Opsonization, helping with phagocytosis. Coating in general, that could be, so neutralization would be, blocking the function of like virus associated with a receptor, whereas coating would be just covering something in general. It won't be able to do anything. Agglutination, having things stick together via association with these antibodies so that they can't perform whatever function they're trying to do. Complement, so going through that complement cascade where we talked about opsonization, inflammation, and cytolysis, and then antitoxin, where the antibody is interacting with what would be the toxin and can't do its function anymore. So our workhorse antibody is IgG, and IgM is always the initial one, and IgA is associated with mucous membranes. IgE is allergies, and IgD is a receptor. So those will start getting produced upon activation of those plasma cells. The primary response with antibody is going to have a latent period. It's going to take a while to get the cells activated and get them producing antibody. And it always starts with IgM, and then we'll shift to IgG. But the secondary or amnesiatic response, where you're refaced with something, that's going to be mostly IgG. It's going to happen very, very quickly. So quickly that you may not even know you were infected or seen the bad guy. Then there's 2 different kinds of basic immunity that you can have. Natural active, which you get immunity from being sick with an illness. Natural passive, that's going to be like breastfeeding. Natural artificial active, that's like introducing a vaccine, so it's a medical procedure. And then artificial passive, like gamma globulin infusions, Humira, and things like that. Vaccines, we talked about the different kinds of vaccines. They do still affect just the same immune system as what we were just talking about. Immunopathology refers to increase or decrease of what your immune system should be doing. Basically, any time that it's abnormal is immunopathology, okay? Hypersensitivity type 1 is allergy or anaphylactic shock. That's going to involve IgE. You'll treat that with antihistamine or epinephrine as you see fit based on whatever reaction is going on. Type 2, we're talking about blood group incompatibility. That's often dealing with ABO blood group as well as Rh factor in hemolytic disease of the newborn for Rh negative mothers and Rh positive babies. In type 3, now we have immune complexes where antibody complexes would be floating around antibody antigen and get stuck in certain areas. This would include lupus. And then we have type 4, which is a delayed type, which involves the T cell response. This includes the tuberculin, PPD, skin test, poison ivy, contact dermatitis, and graft rejection. We did talk about the different kinds of grafts and as far as transplants go. Moving on to the immunodeficiencies, we had primary, which is anytime you have an immunodeficiency from birth. We talked about SCID, which is the bubble boy. And secondary is an acquired immunodeficiency. This would be AIDS or even treatment with radiation or chemotherapy. As far as diagnosing goes, we're going to be talking about, I don't know. I mean, the signs and symptoms, you'll start with your patient. You'll look at the outside, right? The basics of what you have, your signs and symptoms. Then you collect whatever specimens you need. And then you'll analyze like using direct analysis on specimen directly from the patients, or, you might want to culture your specimen first, and you make presumptive data initially, and then confirm that with the confirmatory data to pinpoint the specific identity of the species of whatever's causing the disease. Yeah, so there's different kind of media that people would want to use for cultivation. There's all sorts of different things you can use for biochemical tests, whether it's the media like we used or those long strips like we had talked about. And this all comes back to essentially the same exact thing of that dichotomous key. That really is stuff that they use in hospitals, in the labs anyways. PCR is very, very useful as far as determining, what organism it could be, as long as you have an idea of what route to go down. PCR doesn't decide those things for you. It doesn't run a test against thousands of different things all at once. PCR, you have to basically know what you're targeting when you go into it. So you have to have some idea where you're going with that first. We did talk about antimicrobial sensitivity with Kirby Bauer. Remember, with immunological methods of testing, there is a difference between specificity and selectivity. If something is specific, then as far as what we're talking about with sensitivity, yeah, that's what it's supposed to say, not selectivity, excuse me. I just literally copied and pasted this from the other one, and I remember that I didn't fix it from that one. That's my bad. So specificity versus sensitivity. And sensitivity is referring to, you want to pick up any given positive that could be there, but you might be sacrificing, how specific it is. So you could be having false positives. For specificity, you will not have any false positives. You will only have the positives that are actually positive, but you might not be as sensitive. So you might not pick up all the positives possible. So you might risk a false negative, but that's the only thing that would be the issue there. So that's the differences there. You want to have both, of course, when you're doing your testing. Remember what an ELISA is. We talked about direct versus indirect. Direct is the sandwich method. So you have antibody, antigen, antibody again. So that means you know the antibody. You want to know if the patient has an antigen, and then you're going to retest it back with an antibody linked to an enzyme. Indirect is the antibody there. So you know the antigen. So the antigen is in there. You put that in the well. You see if your patient has the antibody against it. So that's how that would work. Phage typing is using bacteriophages basically as an identifier. So certain bacteriophages infect certain bacteria, and that's that. So you can add in a bacteriophage, and if it is there, if it is able to infect, then you know that it is salmonella or whatever. All right, so this is unit 4. This stuff should be familiar, so I'm going to run through it pretty quickly. Know the different defenses of each of the systems, as well as the biota of the systems. I am not going to review them in this review. So I'm just going to kind of just glaze right over the diseases in each system. So hang in there. Streptococcus pyogenes was associated with impetigo, necrotizing fasciitis, scarlet fever, which was itself associated with it being lysogenized by a phage. Remember, we should know that. I just talked about this less than an hour ago. And then erythrogenic toxin is the toxin that's produced as a result of being lysogenized by that bacteriophage. Rheumatic fever and strep throat, which is going to cause pharyngitis or swollen throat, painful throat, and inflamed. So that's all strep pyogenes. It's not only strep pyogenes that causes these things, but strep pyogenes can be associated with these. Also can be associated with toxic shock syndrome. Staphylococcus aureus, we talked about impetigo and MRSA, with gas gangrene that's caused by Clostridium perfringens. It's also associated with GI food poisoning. Chickenpox is caused by varicella zoster, which is a herpes virus. This is has reactivation version called shingles. Smallpox, this is caused by variola virus, and that would be cause a horrible rash. We've eradicated it from the planet, and the first vaccine was against smallpox. They used vaccinia virus to vaccinate against variola. Measles, rash, it's a virus, get vaccinated. Ringworm, it's fungal, caused by a lot of different fungi. Conjunctivitis can be bacterial, where you would have yellowish or milky discharge. Viral, which you would have a clear exudate, or allergic, which would be a lot of fluid, copious amounts of fluid. Trachoma, this is the leading cause of blindness in the world. This is caused by chlamydia trachomatis, which also causes chlamydia in, sexually transmitted diseases. River blindness is caused by Onchocerca volvulus, which is a roundworm that is associated with having an endosymbiont, which is a bacterium that lives inside of it that makes it more pathogenic. That's Wolbachia. Nervous system diseases. Do you remember that the nervous system has immune privilege? What does that mean? It means that it has a decreased immune response that is protective to the nervous system. Meningitis is a syndrome, right? We have bacterial versions. That would be, the most common would be strep pneumoniae. The worst one is Neisseria meningitidis. That is the meningococcus. It is a gram-negative diplococcus. It also is associated with petechiae like rash. That is very typical of that and isn't found in the other types of meningitis. Haemophilus influenzae and Listeria monocytogenes can also cause bacterial meningitis. The fungal ones that we talked about were Cryptococcus neoformans, which is usually associated with lowered immune function, and Coccidioides immitis, which is valley fever. Viral meningitis is usually caused by enteroviruses that get in through the gut. Meningoencephalitis, that's amoebas, all right? You had the PAM version and you had the GAM version. The PAM is N. fowleri, and acanthamoeba causes GAM. Encephalitis, this is infection and inflammation of the brain itself. That's usually going to be caused by arboviruses. Those are transmitted by ticks or mosquitoes. Poliovirus, we have vaccines available, but it is still active in Afghanistan and Pakistan. It's also an enterovirus. Prions, these are going to be subacute encephalitis. These are infectious protein particles that cause spongiform encephalopathies. They cannot be destroyed with typical autoclaving. They need higher temperatures as well as exposure to sodium hydroxide. Tetanus is caused by Clostridium tetani. Muscles can't relax, and botulism, Clostridium botulinum, muscles can't contract. With cardiovascular diseases, we have blood-based infections. We talked about bacteremia versus septicemia. Bacteremia, any bacteria in the blood. Septicemia, flourishing and growing and infectious in the blood. With the plague, we had that caused by Yersinia pestis. That's a gram-negative bacilli with a reservoir in rodents, primarily prairie dogs here in the United States. It's transmitted by the fleas that bite them. This can be the bubonic, where it grows in the actual lymph nodes themselves, pneumonic in the lungs, or septicemic, which causes sepsis, which leads to the Black Plague due to necrosis of the extremities. Infectious mononucleosis, that's caused by Epstein-Barr, which is a herpes virus, by the way. A lot of them are, it turns out. Epstein-Barr also can cause Burkitt's lymphoma in people who are immunocompromised. That includes people with malaria or AIDS. Malaria itself is caused by the apicomplexan protozoa from Plasmodium, like Plasmodium falciparum, and is transmitted by mosquito bites. Hemorrhagic fevers include Ebola, Marburg, dengue, yellow fever, chikungunya, and Lassa fever. Chagas disease is caused by Trypanosoma cruzi, that bug that bites you and then poops in its bite. They're called triatomes. HIV infects CD4 helper T cells. It has something called reverse transcriptase that turns its little single-stranded RNA genome into a double-stranded DNA that it inserts into the host genome. Respiratory diseases that we talked about, pharyngitis, sore throat, diphtheria, whooping cough. Diphtheria is Corynebacterium diphtheriae. Whooping cough is Bordetella pertussis. Influenza, we talked about hemagglutinin for attachment and neuraminidase. Neuraminidase, again, that was used for budding, so clipping off the sialic acid, attachment to allow the viruses to go free. That is blocked by Tamiflu. That's an example of that. We talked about antigenic drift, which are small mutations over time, gradual ones, and then antigenic shift, where we had the actual genomes getting reassorted in another animal, another host, and that would cause a big change suddenly. And then we talked about pneumonia, which is another syndrome. The most common cause of pneumonia and community-acquired pneumonia, we have Streptococcus pneumoniae. Then we have it, which, by the way, Streptococcus pneumoniae is going to be gram-positive coccus. All right. Viruses will be the next most common. Mycoplasma, the ones that don't have a cell wall. That's walking pneumonia. And then we have randobacteria like Legionella, hantavirus, which is not a bacteria. And then we have Haemophilus influenzae and Histoplasma capsulatum associated typically with immunocompromised. Healthcare associated will be a combination of different bacteria causing it. Common cold could be caused by a ton, 200 different kinds of viruses, okay? Often that can lead to secondary bacterial infections. Tuberculosis is caused by Mycobacterium tuberculosis. We diagnose it with the PPD tuberculin skin test, a chest x-ray, an acid-fast stain of the sputum. You can also have a culture of the sputum and do an acid-fast stain on that. The gastrointestinal diseases, we know how important the gut biome is. The most common infectious disease in people happens to be dental caries. Norovirus and rotavirus are a couple of viruses that cause diarrhea. As far as the bacterial diarrhea go, the most common one is going to be Campylobacter, but we also talked about Shigella, Salmonella, and E. coli, and Clostridium, right? So by Clostridium, I'm talking about Clostridium difficile, so C. diff. Shigella is gram-negative. It's non-motile. It causes dysentery, which is bloody diarrhea. STEC, that is Shigella toxin producing E. coli. So it has the Shigella toxin, the Shiga toxin, excuse me. Same as Shigella has, and it's also gram-negative, right? And they both cause dysentery, so keep that in mind. Moving on, the Enterica and Enteritidis salmonella do not have the typhoid toxin associated with them. The typhi and the typhimurium do have the typhoid toxin, so that's going to determine the pathogenicity of that. Treat diarrhea with fluids and electrolyte replacement. That's the best way to deal with that. Cholera, it's caused by the bacterium Vibrio cholerae, leads to rice water stools. It has an AB enterotoxin that causes the electrolyte release involved with that. It could be deadly due to the dehydration that it causes. Hepatitis is any inflammation of the liver that often leads to jaundice as one of the signs associated with the disease. Hep A is a fecal oral. Hep B, blood and sexual contact. It has reverse transcriptase. It goes from its little DNA genome to its little RNA parts that it works with to make proteins, and then it uses that RNA to make more genome into DNA using reverse transcriptase. Then it puts that into the genome of the hosts to hide out. So C is transmitted through shared blood, so whatever, needle sharing or whatever. There's no vaccine, but antivirals are being successful. As far as the helminths that we talked about, here's some of the basic ones. Pinworms are Enterobius vermicularis. Tapeworms, Tinea solium. Ascaris lumbricoides are the giant intestinal roundworm, which are the most common helminth infection in the world. Hookworms and threadworms through the skin cause itching. Schistosomes also through the skin cause itching. Can be different all over in different areas of the body. Spleen and liver enlargement and can invade the bladder and coat themselves in our blood proteins because it's creepy. Then we talked about the genitourinary diseases in females and versus males. Be aware of differences in biota as well as different things that can cause changes in those biota. UTIs, about 80% of those are going to be caused by E. coli. It's usually the diffusely adherent version of E. coli. Most GI bacteria cannot adhere within the urinary tract because of the desquamation and the flushing of the urine. Right, vaginitis is actual inflammation of the vagina. Vaginosis, infection of the vagina without inflammation of the vagina itself. So vaginitis, Candida albicans, and Trichomonas vaginalis. Then we have vaginosis, that's BV, or bacterial vaginosis. That's going to be caused by a decrease in the lactobacillus bacteria. Gonorrhea is a sexually transmitted disease that's caused by the gonococcus Neisseria gonorrhoeae. Chlamydia is a sexually transmitted disease caused by Chlamydia trachomatis and is the most common of the reportable STIs. Syphilis, we talked about the Tuskegee study, quote unquote, where, undereducated Black men were taken advantage of and forced to go through this 40-year study where they were not treated at all for the illness that they had. Primary syphilis, we have the canker at the site of entry. Secondary syphilis, we have the obvious, very recognizable rash on the palms and the soles of the feet. Also is associated with sore throat, lymphadenopathy, and fever. Tertiary syphilis, we have the development of gummas, which are like soft tissue formations. And then neurosyphilis, which includes blindness and dementia. The most common sexually transmitted infection overall by far is human papillomavirus, or HPV. This can cause genital warts and or cervical cancer, among other types of cancer. Vaccine is protective against quite a few of these strains, often 85 to 95% protective against most things that can be caused by this, and is recommended for adolescents and people who are older, people basically in their sexually active age phases. Herpes, we're talking about herpes simplex virus 1 and 2. Both of these can cause both genital and oral herpes. Okay. Infection with HSV1 or HSV2 does not protect you from the other one. Okay. So if you have HSV1, you're not protected from 2. If you have 2, you're not protected from 1. You can have both of them. Latency will occur with herpes virus into the ganglion of the nerves, and then they will re-emerge and create an outbreak again with stress, any introduction of physical or, even just emotional or mental stress. So that is that as far as the review goes for the final. Jeez, man. Okay. We made it through somehow. So I hope that that was a little bit helpful. I know we skipped over some stuff and there is a lot still that we didn't even cover. Please go back and watch the other reviews for the other units. Look back through the other study guides for the other units. They're going to be more beneficial for you than the final study guide will be, I promise. But if all you have to do all the time that you have is to work on that one study guide, at least that's something, right? So do that at the very minimum and watch this at the bare minimum. And thank you guys for everything and good luck on the final. I hope that you don't come back to my class.